I'm very happy to be here, and I've chatted to some of the patients and their families, so it's great. I think the best part of this experience is also to engage and talk to the patients and families. So I have presented previously in the past, and that's one thing I don't get to do, and I think it reminds me and others in my group the importance of the research they're doing here. And like Lali said in her presentation, I have a form of muscular dystrophy. It's not GNE myopathy, it's limb-girdle muscular dystrophy. So I can bring the angle of not just the science, but also knowing what everyone's going through also in terms of the patients and their families, and also communicate the importance of research. So the research that we do and what I do with Lali and Ralph and Angela in terms of strategizing, we don't want to do research for the sake of research, for the sake of curiosity. It's all about how is this going to help the patients, because I know the patients in this audience have been inconvenienced and been, throughout their journey feel like they're just a guinea pig to satisfy someone else's curiosity. And that's not what I want this to be about, because I've been on the other end of it. And just to let the patients and families know before I present some of the amazing science that, the whole GNE community has contributed to. So the purpose of this presentation is to touch and reiterate and reinforce what Lali said, some of the scientific initiatives that the NDF and the community has supported and where we're going, and also where we need to be. So I want to start off with where we need to be, so that when we present some of the research that's supported this year and the upcoming years, that you understand why each one of these pieces are important for gene therapy and where we need to be. Because when people think about gene therapy, they just think about one part of the process. They don't think the whole picture. Anyone can actually go to clinical trials. Not anyone can actually make a clinical trial successful. And that's something very important that, if you don't get much out of this talk, that you understand that there are many components that come in to make that actually successful. So here's a breakdown of what I'm going to give in terms of the talk. I'm going to talk about a success story and what we can actually learn from that. And then next, a clinical trial that happened in an N=1, so one patient, and what we can actually learn from that too. So it is a form of gene replacement therapy that was performed on a GNE patient, and what we can actually learn from that, and how we can actually build upon it, and from that experience make it better. The state of research and trials at the moment. So a lot of it is a reiteration of what Marianne just presented, but reinforcement is a good thing also. And so I'll take a little bit of a different angle, but she's contributed a lot of that research. So, she would have been the best to present on it. So I'll just lightly go over that again. And then the NDF funded research in the context of all those 3 points, so you understand why we're going in a particular direction. And then I can take questions either during at the end of this talk or during lunch. So I do know I'm the person between you and lunch, so I'll try and stick to time. So I'm going to talk about a successful gene therapy trial in limb-girdle muscular dystrophy. So this involved Jerry Mendell, Dr. Jerry Mendell. And so this was released one day before Rare Disease Day. So that was February the 28th was Rare Disease Day. And a lot of patients, limb-girdle muscular dystrophy patients, were all got up at 8 a.m. in the morning to hear the press release that was broadcast throughout. And that was 8 a.m. East Coast times. So unfortunately, our friends in the West Coast had to get up much earlier than me to actually hear about it. And I'm going to break it down, the announcement, and then try and reflect upon some of the pieces we do have in place and some of the pieces we may not have in place. So limb-girdle muscular dystrophy, the one they're interested in, 2E, is an autosomal recessive muscular dystrophy. So similar to GNE myopathy, you need 2 mutations to cause this disease, one from each parent, so that both copies are defective. And so it's a mutation in a gene called beta-cycloglycan that's found in the cell surface. So unlike GNE myopathy, it's a structural protein that's responsible for maintaining the integrity of the muscle membrane. So patients have difficulties running, jumping, and climbing upstairs before the age of 10. So it's a more rapidly progressing form of muscular dystrophy. You can see loss of ambulation in 10 years, and it often leads to death in age 30. So it's quite progressive, and therefore making a compare and contrast, GNE myopathy is a more slowly progressing disease. So in this case, you should be able to see a benefit much quicker if you pick it at the right time point. So the cohort was of 3 patients between the ages of 4 and 13, so probably much younger than patients for GNE myopathy, and that's the viral vector details, if you can see, and then the dosage. And then after 2 months, they had a look at what the results were. So this was just preliminary results. They didn't actually release any muscle function benefits or anything like that in February because it was too soon. So 2 things I want to highlight is dosage and also safety. So although there were adverse effects, the patients were looked after and things like that. But in order to actually know what those values should be and have a feeling for safety and dosage, they did use a preclinical model, and that's a mouse model, to have a look of, where it was delivered to, and also not just delivered to, but where the protein that they want to deliver to was actually found in terms of the tissue, and also safety. So what was the immune response? So they had a feeling based on using the preclinical model of how they'd actually go about. And also in dosage. So there's one thing to be safe and there's one thing to actually have a benefit. So for example, water is safe, okay, but people wouldn't drink water to address their condition. You also want something that actually works. So, they use the mouse model again to have a look at, do we see a functional benefit, and what dosage would we see a functional benefit in the mouse to guide what we should be doing in a human clinical trial. So the other thing they had is protein biomarkers. So having a biomarker is very, very important. The reason why is if it's a slowly progressing disease, it's really hard to tell initially after, say, a month, 2 months, 3 months, if something is having a functional benefit. We eventually want that to happen, but you want to also have a sneak peek. Everyone wants to know, is it working? Is it working? Is it working? And protein biomarkers, in this case, is a way. So an obvious thing you want to ask is, the protein or the gene we're trying to replace, is it actually in the place where we expect it to be? And so they looked. So the one difference between GNE myopathy and, say, in this case here, we're going from very little protein or no protein to some protein. In GNE myopathy, patients do express the proteins, and so we want to go from no protein to some protein. We want to go from protein with very little function to some function at all, and it's like, how do we go about measuring it? So that's the importance of actually having a biomarker. We can have that sneak peek, know it's actually working in any clinical trial we do. So in this case, because this is a case of going from no protein or very little protein to some protein, they can look at the muscle fibers and ask that question. So they can look at how many muscle fibers actually receive it, what intensity, which is sort of like correlated to how much protein was there, and also they can quantify it also as another way. So again, it's also based on the preclinical model. So looking at the mouse and having a look at how well it actually delivers, what they were actually going to try out in the human also. And also what they could do is also know a little bit about the gene itself and what structures it forms. So beta-sarcoglycan forms a larger structure with all these other proteins, and when you're missing it, the other larger structures can't form. And you can see here, this is alpha-sarcoglycan that's next to it, and dystrophin, which also attaches to that larger structure. And in here, we don't have it, and then when you deliver it, you can see the presence of the other components of the larger structure. So again, we're actually looking for some kind of mark that things are actually happening in the mouse model. So Also, other biomarkers. So CK is a measure of muscle damage. So as your muscle fibers get damaged, CK leaks into in your blood, which then can be measured when a blood draw is taken. So we know in GNE myopathy, this is elevated, but it's more on the mild to moderate compared to other muscular dystrophies. But it's another thing that could be measured during that process, depending on how variable it is in patients. So here, they can see a massive reduction of a muscle damage marker. And this is based, again, on the preclinical model. So a way you can do it in mouse models is actually look at Evans blue dye. So Evans blue dye is sort of like, how it works is just a dye that's, if there's any breaks in a muscle or breaks or leaks in the muscle membrane, this dye can actually diffuse in, so just leak into it. And what you can see here is that red thing here is the dye going in. And what that green represents is muscle fibers that actually have received the protein to be actually expressed. And you can see there is no red in those. So these are protected now from dye leaking in. So there's no damage to those membranes. And so again, that's another measure. So just to summarize, beyond just doing the clinical trial itself, the things that they had in place were ways to quantitate whether it's actually looking at muscle damage, and also having a feel for what was safe and what dose would work based on looking at animal models. So now I'm going to transition to a sort of like a form of gene replacement in GNE myopathy that actually occurred. And this is an N=1 trial, and this was the preclinical data. And you can do the compare and contrast. I'm not going to comment. Okay, so this is another way of delivering a gene to cells in the body is not via virus, which was like in the other example, but is actually through packaging up DNA. Here is the GNE gene on what we call a plasmid. So this is a circular piece of DNA, and packaging it into sort of like a lipid or just like a ball of oil, okay? And this is then able to cross the cell membrane, then get into the cell, and then this gene will be expressed. So a different way of delivering it that doesn't involve a virus. Unfortunately, this way, the field would agree, is not as efficient as delivering to a virus and not as specific to which cells it actually gets to. And this is them demonstrating in this publication the delivery of this piece of DNA, packaging it up in these droplets of oil to cells. So these cells do not have GNE, and here you can see the protein being expressed. But just to emphasize, this is actually delivering it to cells and to cell lines. So these are not particularly any type of cell or anything like that. They're just showing that they can get it into the cell. And here it's showing some kind of a measure, a biomarker here of sialic acid, and the increase of that when they actually deliver it. So this hasn't been done in mouse, so they then transferred to actually doing it in humans. So this was one patient, a 41-year-old male who the disease had progressed in them, and 7 injections of this lipoplex. So it was packaged into that. And then they escalated the dosage across the 7 over a 13-month period. And this is the results, okay? So one, they did show in the quadriceps some kind of expression. It's I wouldn't say it's quantitative, I would say it's qualitative, just to say that we have some expression. And then they also look at sialic acid or an assay, which is an indirect measure of sialic acid in the muscle. And you can see this is not quantitative, it's more qualitative. And for me, it's really hard to actually say whether there is actually a difference before and after treatment here. So again, and then looking at muscle strength, so comparing right to left, and these are the 7 treatments, you can see that it varies a little bit, but it's really, really hard to measure these things. So it's not me saying it's successful or unsuccessful. It's just saying that if we don't have a great way of measuring success, it's really, really hard. And also a great way of knowing what dose would this work in, things like that. So just to summarize, it was only demonstrated on cells and not in an animal model. So, there was no animal model where they showed functional improvement, that the muscles got stronger or something got better. They only showed that they were able to produce that protein in the human cell. Escalating dose on patient because they weren't quite sure which dose would actually work. I would say that the biomarkers that they presented was not quantitative, they were qualitative. And you can see it's very difficult to see if there was actually a difference. And it was difficult to measure the improvement in muscle function. So I want you to reflect upon those two sort of like scenarios, and when I present right at the end some of the goals and initiatives of the NDF for 2019 and beyond, that you understand why that is the case also. Because again, just to reiterate, the goal isn't just to get the clinical trials, it's get to a successful clinical trial. They're two different things. So I'm going to talk about the current state of GNE research. So this is a lot of overlap of what Marion just presented and probably others, but iteration's actually really good. So I would just touch upon all these things. So in terms of the pathology, it's an autosomal recessive. So that means that you have to have two defective copies of this gene, typically one inherited from each one of your parents. It's early to adult onset and slow to moderate progression. So each one of the neuromuscular diseases have a different progression, and something you have to keep in mind in terms of clinical trials and what would be needed to actually show some benefit if something's actually slowly progressing. And it has a different, every one of the neuromuscular diseases have a different muscle wasting pattern. Here it's just highlighted the muscles that are typically affected, but sometimes it does vary a little bit by patient to patient and the severity and when it actually onsets. And the incidence, so Marion has already touched upon this. So the incidence around 1 in a million to 1 in 6 million to 6 in a million, and that varies depending on if we're thinking about founder mutations and what place in the world we're talking about, but typically is around that. And then the onset here, which I already talked about. So in terms of disease pathology, I'll just quickly go over it. So there's a mild increase in CK, which is a marker for muscle damage. And then one of the hallmark features is the rim vacuoles and the protein aggregates you can see in this image. And also it has sort of like its own unique muscle wasting pattern, like a lot of the neuromuscular diseases. So in terms of the gene itself, and one thing I do want to get across, just to reiterate and reinforce what Marion had said, and also what others talking about clinical trials were saying, is that it's very important that each patient gets a definitive diagnosis and a definitive genetic diagnosis, and not just a clinical diagnosis or the clinical presentation. And that means identification of the two mutations that are actually causing your GNE myopathy, and also looking at one actually did indeed come from each one of your parents also, to see that you do have two defective copies instead of two mutations on the same copy of the GNE gene. So one of the challenges beyond looking at the more common ones, which are listed here, is also ones that only occur once. So there's no other patient in the world with that mutation. So sometimes it's really difficult to know if someone is just happens to be a carrier of one mutation, the other one just happens to be a rare variant that does not cause disease. So in genetics terms, this is called a variant of uncertain significance, or a VUS. So you probably typically see it in a genetic report that a lot of the patients are actually sitting on, or have a family member that they've seen their genetic report. So just to touch upon that, and they're there are two functional domains. So they carry out two different tasks in the sialic acid pathway, and these mutations typically line these two critical sort of domains or little areas in the protein that have a responsibility in the pathway of producing sialic acid. So there have been 4 mouse models that have been created. One common feature between each of the mouse models is the reduced survivability. So they typically die early in life. How early, depending on what mouse model and what mutation. So these are from the Japanese group, from the 2 mutations commonly found in Japanese patients, and these model a mutation commonly found in the Persian Jewish community and also in the Middle East. And one of the most challenging things about the mouse model is that either they don't have a muscle phenotype, which is unfortunate because a lot of the people in this room, GNE myopathy, do have something wrong with their muscles. So the other thing is they don't have a reliable muscle phenotype. So even those that have a muscle disease, sometimes we're unable to reproduce that. And so it is one of the challenges in the field at the moment that if we are going to try therapeutics and we want to know what its benefits are, not in terms of sialic acid, but what benefits does it have in terms of muscle function? At the moment, we don't have the best animal model possible. We can use these animal models, but there is a lot of room for improvement. So in terms of the disease mechanism, so GNE is responsible for is a critical protein and enzyme in the sialic acid pathway. And that's why, as Marion presented, MANAC and supplementation by a derivative of sialic acid is actually a rational form of therapy. So if it can't make this, well, why can't we supplement the cells with these things that it can't make? And one of the features is hyposialation, so a reduced amount of sialic acid. And here is looking at biomarkers in a muscle biopsy here, and you can see the reduced levels in the GNE patient. So again, there's still a long way to go in improving the quantification of these biomarkers also. So using that sort of like knowledge and sort of like pathway to rational therapy, and this is the motivation of some of the MANAC work that has been presented and trials have been presented, and also sialic acid supplementation. So it was shown in this publication here in Nature Medicine by the Japanese group that supplementation, you can reverse some of the muscle phenotype, and these are some of the measures of the reversal of that. And others have shown in their mouse model in different ways that of improvement also. So one of the things that they saw is sort of like an increase in survivability also with this supplementation. So, and that's the motivation to the Ultragenyx clinical trial. And one of the big things we got out of a lot of the clinical trials is not just sort of like a rational therapy for patients, but also clinical outcome measures. What should we be measuring in clinical trials? And also we have a wealth of data now of how patients, progress over those months that they're on the clinical trial. So, in a way, we indirectly got a natural history also as those patients came in and we took those measurements on them. So sort of like a bunch of sort of like measurements and functional outcomes were established in a lot of these clinical trials that we can then use in future clinical trials also, which I'm pretty sure are being used. So that's one of the big things. And so here's a summary in this publication by Nuria of the two main strategies in terms of clinical trials for GNE myopathy. One is replacement of things that GNE can't make and supplementing the body with it. And the other approach is supplementing the gene itself, the gene or the protein itself. So they're the two sort of like rational main approaches you can take in treating this disease. So another thing that's been happening both at the NIH and also at Newcastle University is a patient registry and a natural history study, and you can see the number of patients involved in each, and this is just taking functional measurements over time. And what this has allowed, say, the NIH group to do is to actually create predictive models of what Marion mentioned as disease age. So to know what equivalent this person is based on data from natural history studies and how they're progressing, what their disease age is, because everyone progresses at a slightly different rate and things like that. And what the Newcastle group has used their natural history study to do is look at genotype-phenotype relationship. So based on your mutation, are you more likely to progress faster? Is your disease more severe and things like that? Because they had a wealth of information on each one of their patients, including the mutation that each patient has. In terms of the preclinical work, so Stella and working with the group in Columbus had worked on a adeno-associated virus that they published here. But again, just remember that there are no good mouse models with a reliable, reproducible muscle phenotype. So one thing they did demonstrate in this study is safety. So, and also delivery. So does it get to every organ in the body, at what levels, and what's actually being expressed? So they were able to demonstrate some kind of safety and proof of concept. But the ultimate thing is showing muscle function benefits also. But unfortunately, we don't have the best animal model to actually show that at the moment. So in summary, we still lack a good animal model with reproducible muscle phenotype required to validate some of the therapies. So there's a lack of complete understanding of GNE genetics. So, and also that protein may have function beyond the sialic acid pathway. And it shows that we have to go back and do, constantly be doing basic research, looking at the pathways too, because if the GNE gene had more function other than being involved in the sialic acid pathway, we may be only addressing part of the problem and not the full problem. So we know we are addressing some of the problem, but we're always wondering, could we do better? With supplementing with something else also on top of that in combination. So if it does have more function, then we have to go back and do the basic research to actually have a look at that. And also the further development of quantitative biomarkers. We want to know, and we want to know early if something is working, in whether it be the mouse model or also in the human clinical trial. And yes, we do have biomarkers, but there's always room for improvement and, improvement in sensitivity and specificity. So these are some of the NDF funded research for 2019. And hopefully I can link to how this will contribute to the increased success of future clinical trials. So first is the patient registry. So to know what patients are out there and what mutations they have, and just the most basic details, so we can then get some insight. So this was led by Amy and Tara. So their hard work of collecting approximately 200 patients so far, and Danielle, who did a brief spring internship with us. And one thing you can do when you sit on patient registry data is you can look at weaknesses on where we can improve. And one notable thing about patient registry and asking for people to volunteer, the females actually do a lot better at volunteering than males. So when Danielle went through it, I'm going, I bet you if you do a proportion of male to female, you'll see it won't be a 50-50, even though this is not a a sex-linked disease or things like this. This is just typically how it goes with a lot of things that are volunteer-based. So if there are guys that are listening or in this audience, do encourage your fellow male patients to volunteer and contribute to this, because this should be closer to 50-50. And if I was going to take a wild guess, the unknown are probably mostly female also. So for the guys out there, don't pat yourself on the back up. Oh, the unknown are probably male. But I think a large proportion of that is that. So, we'd like to get this closer to 50-50. So this is not a criticism of Amy and Tara's efforts. This is criticism of male patients trying to pick up their game on this. And here is a breakdown of where these patients are in just the United States. So I'm going to go back and work with Danielle to do a scaled plot, because here I'm just showing where they are, but not the circles aren't proportional how many patients in each particular city. So I'm pretty sure we have good representation of the people right here that are in the registry on the East Coast. So thank you very much for, coming to this wonderful event. And so again, with this registry, it shows, some of the gaps. So you can see that there are probably patients in the middle of America and things like that. So it allows us to see where the gaps are. And you can see that the coasts are well represented, and a little bit inside. And here, of course, I think it's Columbus, Ohio also, so we're not showing where the patients are living, we're showing where they're going for clinical trials or going to see a doctor, because patients move all the time, but they typically stick to the same doctor, even if it involves a little bit of travel. So here we're just showing where they're going to see their doctor. But here I'm showing the benefits of registry, and in terms of knowing where our our areas of improvement are. So that's just showing a little bit of snapshot. So one thing others on the board asked me to spend a little bit of time on is trying to differentiate the whole genome sequencing project and the biobanking project. So the whole genome sequencing project is an initiative that was kicked off about a year and a half ago, or a little bit earlier than that. And that was to characterize the genetics of GNE myopathy. So it's to recruit patients that already have a genetic diagnosis, not trying to get them a genetic diagnosis, but it's the fully characterized genetics. So what are the genetic variants that we're finding in their genome that could either contribute to the disease? So talking to some patients this morning, it's a known fact, and it's a known fact for a lot of neuromuscular diseases, that it affects family members who have the disease a little bit differently. So some people get it earlier than others, some are more severe than others, some it progresses more. And it's to ask the question, is there anything in terms of the genetics of each one of those family members that may be contributing to it? It may be underpowered, but it's the start of asking that questions to pick up anything that's obvious. So, and it's a way of us going to other researchers. Well, we did the study on 100 patients. We either found something or we didn't find something, and at least we can close that book. And it's also, one interesting thing is looking at people who have mutations but don't have the disease yet, but they're in their 60s, 70s, and 80s. So, what is it behind this genetic mystery? Is there some sort of like genetic modifier that's making this disease less severe or later onset, and what can we actually learn from that? Or is it just, or we don't have enough data? But it is an interesting area to explore also, because there are some patients out there that fit under that category. If you're one of them or one of them, do let Lali and Nancy know, and others. So a pilot study has been completed in collaboration with the NIH, and that's 8 samples from 2 families. And we decided to do these 2 families because they had already had exome sequencing. So that's just the coding parts of their gene. So not the whole genome, but 1% of the genome had been sequenced. So it allows us to do a lot of quality control in terms of moving forwards, because it's family members. We can see how the variants are inherited and things like that. So it's a way of just sort of like having these checks and measures on that project. At the moment, the study is small, so the current analysis have revealed no new findings, but but we're going to be building upon it. And that's why PerkinElmer are here to collect saliva to go to this effort. So we really would like people from larger families and also smaller families to contribute, and we can then pick and decide which ones are actually going to increase our knowledge in terms of the genetics of GNE myopathy. So this is separate to the biobank, and you'll hear about the biobanking effort also in a moment. So another funded effort, if you don't remember any of the stuff I said during the 1 hour, is that we do need to move towards a better animal model. We do have some animal models, but there's always room for improvement. And one of the things that we can't create, typically in genetic studies, we'll create a knockout mouse model. So a knockout mouse model is to completely remove the gene or the gene's function in the mouse. And unfortunately, we can't do that with GNE myopathy because GNE or some functional GNE is required for survival and for life. So you can't create a mouse that completely does not have GNE, and we do not have any patients out there that have complete loss of GNE or loss of function of GNE. So one way we can actually, compromise on that is to create a conditional knockout. So that means removing the gene's function, completely removing the gene's function in a set of tissues at a particular time in development also. So that way it can get past the survivability hump, and then we can get rid of the gene somewhere. So it's the hope that if we say get rid of the gene in the muscle, for example, that we'll start seeing muscle weakness in the mice. And then we can then try our therapies to ask the question of, does our therapies improve that muscle deficiency we see in mice? The other thing that Stella's group is working on is also a zebrafish model. One benefit of the zebrafish model is you can rapidly screen drugs and prioritize drugs that look promising that then you could try in a mouse model, which is a lot slower to try drugs, but a lot more closer to a human. But zebrafish models allow you to rapidly screen drugs. So Stella's group is also working on creating a zebrafish model of GNE that has some of the features of the disease itself that she can look for improvements as she rapidly screens drugs or different therapeutic approaches. Next is biomarkers. So one of the things that may say GNE distinct from a lot of the neuromuscular diseases, it's mode of action. As I said, that you can't have complete loss of this protein, unlike other muscular dystrophies. So sometimes measuring the protein levels is not genetically correct. It should be measuring the function of the protein rather than the levels. But having said that, unfortunately, there is no reliable antibody at the moment for the GNE protein, so a way of measuring. But there hasn't been sort of like a priority because of the mechanism of we're not looking for levels, but it'd be great to have an antibody regardless. So as I said, mutations do not necessarily reduce the levels, so it's more impacting on its function. And that's why measuring biomarkers such as its impact on silation and other indirect measures of its function is very important, because that will then be sort of like the surrogate biomarker other than looking at, an indirect measure of the protein function, because we can't look at levels. And so this is so one of the efforts is Marianne's group looking at that, among other efforts from the NIH, which you've seen Marianne present. Another effort is the biobanking. So the biobanking was an initiative launched at UCLA at a patient conference, and we were able to collect 9 patient samples and from skin biopsies and 6 other family members. And what this allows us to do is now create a resource for the GNE research community to look more into the mechanisms. Like I touched before, if GNE had other functions or we want to actually create more reliable biomarkers, having access to patient samples allows people to do that, and then be able to predict what would happen in a clinical trial if we had some samples like this. So we've established this at Yale, and it's contained at Yale, but it's not for us to hoard and keep. This is for us to share with the GNE research community. So we've already shared it with Noah at Ohio State University, who wants to do some various assays that his group specializes in terms of muscle function, and also Kelly Harden, who will talk a little bit about some of her plans in terms of looking at biomarkers, which is a good transition to some of the work that Kelly got funded for in terms of using some of the patient samples to improve on biomarkers, and also look for other biomarkers that with her knowledge of the sialic acid pathway. And she'll talk later on about that, so I won't go into more detail of that. Also, the biobanking allows us to do other things. It allows us to look at other ways we could correct the gene mutation itself. And so one method that has only become available in the last 2 to 3 years, mainly around 2 years, is being able to go to the mutation, say the Persian Jewish mutation, and just change that one letter of the DNA in the genome with that precision. So it's different than gene replacement. Gene replacement is giving you a functional copy of GNE, while this is just going to your DNA and just switching. So just changing the typo in your DNA, that one letter typo that in patients that are giving their disease. So Catherine will be touching upon it. So it's just to show proof of concept. Can we, from some of the patient cells that were collected either at the UCLA event and today's event, can we actually reverse the DNA change, reverse that mutation that's causing your disease? And so, I think it's a very promising approach, and it's sort of like something that will work in parallel with gene replacement. The other thing it will allow is isogenic control. And what I mean by isogenic control is when you work on therapies and on cells and you're comparing two cells, on average, there are millions of changes in the DNA between the two cells. And what that means is that there's a lot of variability, genetic variability. There are other things that contribute to variability, but scientists in the crowd that have worked with cells from patients know about this variability. And what an isogenic control is if we can correct the patient mutation, that will be then we'll have two cells. We'll have one from the patient with the mutation and one from the patient without the mutation. And that will be the only difference between the two cells. So we're getting rid of some variability that now we can control, and know that if we do apply therapeutic approaches, or we do look at mechanism, that we've reduced the variability to the best of our ability. And they are the error bars that contribute to, say, things like this. You can see that variability. And so that's one way of reducing that variability and getting statistical significance and being more sure of some of the results we're getting. So in summary, these are the NDF fund research, and hopefully I've touched upon why each one of these are important for our goal for a successful clinical trial, for future successful clinical trials, including gene therapy, and how it will add to the wealth of knowledge that we've already built upon in terms of the GNE myopathy field. So thank you, and I'm happy to take questions.
Thank you so much. Thank you so much for that very, very informative talk. Monkol, I know you're tired standing out there, I'm certain of it. If there are any questions for Monkol, please let me know. But we do have, while people are thinking about coming up with a question, I want to reiterate the importance of patient sample donation and information to Amy, Curran, and or our other CPA out there, who I'm going to have come up for a few minutes after you are done. Yep. But there should be, in your humble opinion, should every patient here today do you think, is it important, yourself as a patient, being sensitive to that topic? It really is important for us to have as much information as possible. So skin, blood, tissue, everyone's being wonderful and going out and doing that. But at the very least, at least saliva. And for affected family members, unaffected family members in 3 generations. I know you said that, and I don't want to put too fine a point on it, but we really do want to rally people to do that. And can you talk a little bit about what we're collecting for the whole genomic sequencing that's going to PerkinElmer versus what you're doing, and how that will converge in the database?
Yep. So I'll just reiterate on that point. So so the things that are being collected for the PerkinElmer study is we're trying to hit 100 patients, and that is to look at the genetics and trying to further our understanding the genetics of GNE myopathy. So that's as I said in the talk, looking at other things that could contribute to the severity of the disease, the onset of the disease, and just to be 100% sure that there's nothing else that we should be aware of, particularly when we're recruiting patients in the future for clinical trials and things like that, because as the Newcastle group has shown that there is some kind of genotype-phenotype relationship. So what mutation you have sometimes has an effect on the severity of the disease and things like that. But as we gather more data, we'll know whether that is just variability or whether that is sort of like the case also. While the biobanking efforts is more to understand the mechanism, and also the mechanism of disease, and also future biomarkers, which are sort of like interrelated, one and the same. Having patient cells allows us, as Catherine will present, allows us to get this once from you and not have to bother you again, because from those cells, we can also get DNA and other things too. While the collection of blood for the biobanking allows for 2 things. One, if we preserve it correctly, we can look for biomarkers in the blood. So anything we discover in the muscle or the skin cells and things like that, we may have an option to have a look at, is it also in the blood? Is the signal good enough to pick up? Because one of the goals is not just to get biomarkers, but to get biomarkers that are minimally invasive and it doesn't hurt the patients, because I've given a muscle biopsy before, I've given a skin biopsy before. I'd rather just have a prick of my blood or a bit of my blood out of all 3 of them. And so it's to cover all bases. The other reason why we're collecting blood samples for the biobanking is future proofing, because collection of saliva DNA and DNA now creates it doesn't matter if you don't know the technical reason for this, it creates fragmented DNA. Newer technologies can make use of higher quality DNA that are longer and not fragmented. So it's future proofing. If there are new technologies to look at long length DNA that could contribute to the genetics of GNE myopathy, we want to future proof and extract the DNA at its highest quality.
Thank you for that answer. We have a family here I'd like to introduce you to, and I think they may have already spoken to you, but and this is my own non-scientific question, they have a son who's 14 years old, who's recently been diagnosed a year ago. I'll let you talk about that in a second. But one of the things that is interesting to me as an outside observer is the fact that he's already symptomatic at this age, which is something we haven't really seen before. Could it be possible that he has a dual diagnosis, and how will his DNA help us determine if maybe there's something else going on as well? I'm going to pose that question, but introduce you to Mom, who wants to maybe tell us a little bit about her son.
Hi, I'm Tammy. Rich and I have a son, William, and he's 14, and when he was born, he had a platelet issue. He did have some low muscle tone, and that kind of stayed normal, for years. And then maybe, I want to say 4 years ago, maybe 3, his neuromuscular doctor noticed that he was having some atrophy in his lower extremities. She did some testing, and he was losing reflexes in his ankle. So they sent away for the exome sequencing, and I guess we got the results back maybe 2 years ago. He had two, mutations in the GNE gene, one for myself and one for my husband. And anyway, we're just, I've been in touch with the NIH. I don't know if he can be in any of the MANAC trials. He is 14, but we're just trying to, get some help from him. We're trying to get the whole genome sequencing for him. We're having trouble with that. So I don't know. He's an outlier, in the GNE. So he doesn't present typically. So I don't know what's going on really,
Yep.
So to follow up on my question, is any of the sample taking that we're doing going to be able to give us more information, or are there secondary tests required of everyone as we're looking at the role of modifiers or anything additional to GNE? Can you address that, or is that a question more for, like, say, Dr. Paul Martin? Or I really.
Well, I can try and address in terms of the genetics of and also the possibility, but this is just a possibility of having two genetic diseases and things like that. It is very unfortunate, but the most unfortunate story I've heard is someone who had both Duchenne muscular dystrophy and cystic fibrosis. So, it is a very, very rare probability. But having said, there is a possibility of having two separate genetic diseases, one to do with the platelets, one to do with GNE myopathy. The other challenge is, as I touched upon, is variants of uncertain significance. So a lot of patients have a mutation that's common to their region of the world where their ancestry comes from, and sometimes that's in combination with a variant that's never ever seen before. So and looking at the genetic report, one was classified as a variant of uncertain significance. And it brings up a point, and I'm not the best person to answer that, but in terms of clinical trials, whether a person with either one variant of uncertain significance or two variants of uncertain significance would qualify for clinical trials. And it's important for patients to know this because the previous speaker did touch upon having a genetic diagnosis, but there's also having a genetic diagnosis in the gray area, also of having the variants of uncertain significance, and whether having one or having one is still okay if the other one is pathogenic, and would it be okay to have two variants of uncertain significance for these clinical trials that rely on a genetic diagnosis? So it's just a point of discussion for people who run these clinical trials, which I don't, but I more look at the genetics of it. So doing whole genome sequencing is not going to improve that state. So if something is a variant of uncertain significance, it doesn't mean we have a problem in detecting it. It means we have a problem of interpreting whether it actually causes disease. If we do whole genome sequencing, we will still see that same variant, fingers crossed, but 99.9% of the time, if samples aren't mislabeled, we will see it again, and we will still have that same problem of interpretation. That's why work that, say, Kelly, Marianne, and other research, and Stella and other researchers, part of the GNE research community, their research is so important to understand the mechanism of the disease, because then it allows us to develop other assays that are more informative and correlate with disease, so that things that are classified as variant of uncertain significance, we can be more certain if it actually causes disease. Because as the previous speaker was saying, that if we're not 100% sure it's GNE myopathy, then MANAC may not have any effect in that scenario also. And that's why it's important in terms of the genetics to actually have a wider understanding of the genetics, including variants of uncertain significance. But where whole genome sequencing could help is also where we only have one pathogenic variant, and the variant of uncertain significance is a marker of something else that previous genetic testing couldn't actually show, but whole genome sequencing does show.
And has your son, in fact, provided samples, tissue sample or blood sample?
Yes, he provided a muscle biopsy. Yes, we provided a muscle biopsy to Dr. Hegde and her team. And they wanted the whole genome sequencing, but we haven't done that yet. But I have a question. You know, that article that I gave to you about the 9 GNE patients that had macro thrombocytopenia, including my son, would they be able to use a drug on those patients to see if that would actually boost the platelets up? Wouldn't that be an easy way to look at things to just in that regard? And maybe that would help say for my son that yes, he does have GNE. Do you know what I'm saying? Because they were saying that the platelet issue is from the silicic acid in these 9 patients.
So unfortunately, I can't answer that question because it's outside my expertise. So sorry.
Well, I think later on today we're going to hear from both Dr. Paul Martin and Dr. Kelly Harden, and perhaps we can shed a little bit more light on how gene therapy may or may not be an appropriate treatment course for your son. I mean, again, this is an uninformed question, but Dr. Martin, could you answer the question that wouldn't a 14-year-old be a good candidate if he were just a GNE myopathy patient? Would he make a good candidate for gene therapy?
It's impossible to say definitively.
He says it's impossible to say.
But certainly, in all of our experience with gene therapy with progressive diseases, the younger patients are treated, the more effective those therapies are. So, in that sense, yes.
Thank you for offering some support. Is there any other question for Monkol? Otherwise, Angela, maybe you want to help Monkol down from the podium.
I think there's one more question.
Oh, there's somebody raising their hand, but Angela, you still might want to hover over there. Who was it who had the question? Imad? Okay. One moment.
Thank you for the presentation, Doctor. So I had a number of questions, but I'm going to save like half of them for lunchtime.
Yep.
But in your presentation, you mentioned about, or not just you, but rather everyone has so far mentioned about this, the quadriceps sparing. I'm a little critical about it, just from the sake of how I experience my own symptoms. Yep. I feel like, is it true that could it be just affecting the quadriceps less compared to the other muscles, or maybe slower as the disease age progresses? So that's question number 1. Question number 2, you mentioned the importance of getting a definitive diagnosis rather than just a clinical one. Yeah. And you also mentioned something there that you can have 2 mutations where it may not necessarily be coming from each of the parents, but rather could be one random mutation and one from one of the parents. I'm just not really sure how that works, but I would like to understand that you also mentioned how it's important for like the parents to also confirm their mutations, or whether or not they're carriers and things like that. Are we doing something like that at NDF, where we're genetically testing parents and other family members across the board?
Okay, so I'll try and answer everything I can remember. So the first thing about the muscle wasting patterns and things like that, unfortunately, I'm not the best person to ask because that would be a physician that routinely sees patients and performs MRI. But one of the things we know that whatever is published is kind of generalizing and just summarizing the majority of patients, because if it didn't, then it would have to be a lot of accept this and accept that. So, it's not saying that all patients are that way, but I can't comment further because I'm not a clinician that looks at patient MRIs and looking at their muscle wasting pattern. But the second point is a very important question point that you bring up, that it is important for recessive diseases to look at what we call the segregation pattern, that that both copies are defective. And so we call it in genetics whether something is in cis or in trans. So if it's in cis, it's mutations on the same gene, while if it's in trans, it's in the two copies of the genes or the two different chromosomes that you inherit from your parents. And typically genetic testing for recessive diseases, they typically would just be the patient, and then afterwards they would request for the parents to also get tested. And when they test that, they only test for that variant so that each one of your parents are a carrier. But there is a scenario where you just said, but this is very rare for recessive diseases, where one parent's a carrier and one parent is not a carrier. So one parent is a carrier of one of the mutations, not both the mutations, but one of the mutations. The other parent is a carrier, not a carrier of any of the mutations. And so in fact, it's actually what we call a de novo mutation. So a new mutation in one of the parents that was not in their blood cells in the body where they got the DNA, but rather in their sperm or egg that got passed on to the next generation. That rarely happens in recessive diseases. It more happens in dominant diseases, what we call de novo, but it does happen. But it's important to actually know that, and most genetic testing will do that. They will call in the parents to actually look at that, because it's genetics 101 to actually check the parents also. But sometimes it is hard because life is life, and sometimes people don't talk to their parents when they become adults, and it's hard to track down the parents, or people get diagnosed later on in life in their 40s and 50s, and then their parents, sadly one of their parents have passed away or things like that, and we can't make that confirmation. So sometimes that's the best you can go with, because life's life and you can't get the DNA from the parents.
But with regards to the portion of his question that we are at NDF attempting to take samples from all generations, the patient, siblings, parents, many of you here have done this today, and any offspring, because that really then gives us a full picture, right?
But just to confirm that what the NDF is doing for these scientific initiatives and endeavors is not clinical sequencing. So it's not to find anyone's genetic diagnosis. So the work we do at Yale and work that other collaborators do, the only reason why they would sequence over a mutation is to validate. It's to validate the samples who we think it's from, based on the mutation they reported, that they are actually in the cells. There weren't mislabeling, there wasn't any mishandling in the process. It's not to actually give them a genetic diagnosis. So it's only confirming what is known, and it never gets communicated to the patient. So hopefully that is clarified. PerkinElmer and other, clinical sequencing providers actually provide that service via your physician.
Thank you so much for that wonderful talk, Monkol. Please give a round of applause for Monkol.